Hey guys, this is D. So in today's video we're gonna go over information for the exam on Thursday. So it covers pretty much chapter 1 through 6, and what I'm gonna do is I'm gonna go over the questions and kind of a detail where they correlate with one another instead of being separate topics, so you'll just understand how they work as a collective unit. I think that's just more helpful prior to the exam. I have a whiteboard instead of pieces of paper, so it might be more helpful and clearer to see. So just let me know, okay? So the first thing he asked was, what are molecules? So if you remember from chapter 1, you have elements. Elements vary from hydrogen, sodium. I'm just naming a few, so these are kind of like the essential nutrients. You have potassium, you have calcium, oxygen, radon, boron, etc. So the circle is going to stand for hydrogen at least. And hydrogen has 1 electron. That's why it's number 1. Remember, elements and atoms kind of coexist by themselves, but the way that we use them in our body doesn't necessarily go that way. So we use them as molecules. The reason why we use them as molecules is due to abundance reasons. So for instance, in H2O or water, you'll have 1 molecule of oxygen, 2 molecules of hydrogen. And what's important about that is that the hydrogen molecule in water is twofold. So any atom that comes in 2 or more in nature is considered a molecule. Next thing we want to understand is neutral, positive ions. What makes a ion positive, what makes it negative, what makes it neutral. So we'll stick with water for now. So based on the diagram in the book, we had delta, minus, positive. So then when we draw another water molecule, you'll have the hydrogen attracting. To that other water molecule. So then what's important is going to be electron distribution between molecules. So as I said again, we understood that molecules are atoms that exist naturally in pairs and triplets, etc. So for instance, you want to have hydrogen molecules, H2, O2, hydronium, H3O plus. You'll have magnesium as an ion instead of as just a molecule, because you get a lot of magnesium from food. So you can get molecules and their ions from food sources as well. Next thing that's important that kind of connects them together is that electron sharing happens between two atoms or two molecules. So for instance, we're going to use Na for sodium as in the textbook, and Cl chloride. Alright, so what's most important about this is that sodium and chloride both, they don't like to be by themselves. So you're naturally not going to find chloride and sodium alone. So you have Na, you have the proton and the neutron. That's to mix up the weight of the atom itself on the periodic table. And then you have rings. I prefer not to call them rings, but I prefer to call them clouds based on what they actually look like in real life. Electron clouds. And these electron clouds have electrons. So I'm going to mark them off by X. So in the first ring, we're going to have 2. Same thing for chloride there. Then you're going to have 6. And then you're supposed to have let's see here. Do this right. Alright, so Na has 1, so sodium has 1 on the valence, and then chloride has 1, 2, 3, 4, 5, 6, has 7. So in order for chlorine to be happy, it has to accept the electron in the valence shell, which is the outermost, bring it over to chlorine so that it can fill its valence shell. So that's how sodium chlorine forms. So when we provide solutions via IV sodium chloride, be given it a different concentration of the solute sodium to aqueous or water-based solution. Solvent. Sorry. So, then from there, what we want to understand is that H plus or hydrogen molecules or ions. So H plus gives us a pH scale. Alright? So maximum is 14, lowest is 1. 14 is basic, 1 is very acidic. Okay? So what that really means is regarding to like molecules and ions and things like that, is that the more hydrogen is in a particular molecule, the more acidic it is. The more hydrogen ions is in a molecule, the more acidic that is, such as lactic acid. The more basic is the less hydrogen, and you'll have like more hydroxide. You'll have more hydronium as well. Excuse me. That's what's important there. So drawing all three concepts together, you'll have electrons being given up by sodium, that makes it more positive than it originally was. So in doing so, it makes it a cation. By chlorine accepting the electron, making it more negative, it becomes an anion. By putting a lot of sodium into water, you're making the concentration of solute to solvent very high, meaning a lot of salt crystals compared to water or the environment that it lives in. And then as far as pH, it's not going to be as acidic anymore. Okay? So then from there, we want to understand what cells do with it. So what cells would do, cell would see NaCl. So you see a lot of salt outside of the cell. So you have little organelles. Cell membrane. You see the cell outside. The cell will either take it in or excrete it out. The taking in is pinocytosis or cell drinking, which is a form of endocytosis. The taking out is a form of exocytosis or expulsion. Okay? And this is all going to be either in an aqueous solution, water, or in blood. So that leads us to the next question, which is going to be, what is included in the cell structure? So what makes the cell able to endocytose and exocytose are organelles. So, understanding what ions and molecules and all that jazz is about, we can move on to the structures of the cell. So for one, you have the mitochondria, the powerhouse. We'll come back to that later. Which has a intra and extracellular matrix. You have the lysosome, which have enzymes that break down nutrients and other biochemical substances inside of the cell and also aid in cell death, programmed cell death. You have the Golgi apparatus, which is kind of shipping and posting within the cell. So you can call it a post office. The reason why is because it usually encapsulates things in vesicles. Okay? So whether it's the cell taking in the sodium or taking in a protein or taking in a primary messenger such as epinephrine, the cell is now understanding the makeup of that messenger or the makeup of that nutrient. So based on the concentration of ions, the cell is going to understand the difference whether it's too acidic for the cell, it's too basic for the cell, or in other words, it's too alkalotic, which is basic, or it's too acidic or acidotic for the cell. Okay? So the Golgi also helps with that because it kind of ensures via little messages that the Golgi sends out, as well as when it accepts things in, whether or not it's stamped for approval for the cell to use as nutrient, use as food. And the mitochondria, since it has its own DNA, can also do that as well by sending out proteins. You have the endoplasmic reticulum, the RER, and the SER. The RER is called rough, and the SER is called smooth, simply due to the presence and therefore absence of ribosomes. And these aid in protein synthesis. Okay? So that little line there is, say for instance, mRNA, and you get the two ribosomal subunits. So again, the mRNA, the nucleotides, or the base pairs, also have acidic basic components to them. So the reason why is ribonucleic acid instead of ribonucleic base is due to hydrogen content or hydrogen ion content. So that all relates back to molecules, ions, atoms, and how they relate to one another. Moving on up, basically you'll have the nucleus of the cell, contains DNA tightly bounded on chromosomes. Or mitosis, which just kind of looks like sister chromatids. Alright? Move right along. So how does this all kind of matter in terms of molecules, ions, parts of the cell, cell drinking and eating based on protein synthesis as well as receptors and things like that in terms of the human body? Well, it helps with digestion, which is a form of metabolism. It aids in tissue production, protein production, what a tissue ultimately becomes based on DNA programmed by the cells, mitotic versus meiotic factors, as well as whether it's a cell that prefers a very acidotic or very alkalotic environment. So to take that one step further, we're going to go on to the question that says, what is the end product of mitosis, as well as what is the reaction or digestion of food would be, and as well as let's put in another question, the cell cycle, the Krebs cycle. So alright, so we understand that we have acids and bases. We understand molecules. We understand the cell functions a certain way to create proteins, etc. But how does it get the energy to do this? Does it through the TCA or the Krebs cycle or citric acid cycle? And I want to pay more attention to the citric acid cycle name because of the acidotic part of it. So you have nutrients in the form of amino acids. You have nutrients in the form of lipids or fats, as well as having carbohydrates or sugars. These are different forms of energy that your body gets from digestion. And then from there, what your body does with it is that it either converts it to pyruvate or acetylcoenzyme A. And that basically starts energy production downstream in the mitochondria. So for amino acids, which comes from or is derived from proteins, they turn into pyruvate. So this is in your book and from lecture. And lipids come from triglycerides, which have the glycerol backbone, and donate electrons just by nature. And the carbs, simple sugars, come down into pyruvate and acetylcoenzyme A. And then that starts the Krebs cycle with the presence of oxygen. So all of this going into acetylcoenzyme A, etc., doesn't happen readily without the presence of oxygen to kind of even things out. So facultative anaerobes, or prokaryotes, for instance, do not readily have a lot of oxygen predisposed to them. Or a guy in the gym, for instance, who's working out really hard at a bench press machine, or who's at a leg press machine, is working really hard to produce energy, but he's losing oxygen. He's also using ATP. He's also burning sugars and possibly burning fat. Okay? So you have our guy there, and because the body doesn't have the oxygen it needs to continue making lots of ATP, which is at the end of the Krebs cycle, you're making 36. Instead, it gets converted to lactic acid. So lactic acid, little or no O2, molecule of oxygen. Okay? Moving along nice and swimmingly. The main takeaway from that is that with oxygen, you're creating a lot more energy. Without oxygen, you're getting lactic acid and you're burning a lot of fat now instead of sugar as fuel. So when your body repairs now, and it's the next day, and you have micro tears in your muscle cells, it undergoes mitotic division instead of meiotic division, and you end up from one and this is in the textbook as well, end up with 4. Okay? And this keeps going and going and going and going, but it's of the same genetic material as the original cell. So now that your muscles are filled, you'll undergo a mitotic division. What is a muscle cell? How do you have muscles? What is muscle tissue? So histology, or the study of tissue, with regards to muscle cells, is due to one little stem cell now differentiating into other cells. So for the muscle cell, muscles, at least depending on your blood type for instance, like to be more basic based on in much more need of blood compared to other cells of the body than acidic. So it ties back to molecules, content, hydrogen ion content, etc. So for muscle cells, they're striated, so they have multiple layers, they have nuclei, these are Z fibers, you have myosin and actin, and the cells from differentiation and based on being in a more alkalotic environment, they build this nice striated structure or layered structure. Okay, for muscle cells, once they've contributed and amassed a certain amount, they become a tissue. So you have element atom, molecule, compound. Then you have an arrangement of compounds, proteins creating structures. You have cells. Then you have cells coming together to form tissue, so in this case muscle tissue, based on whether or not they like acid, base, whether they're located ventral in the front or dorsal in the back of your body, whether it's in an acidic environment such as with hydrochloric acid formation, or in the lungs where it's a lot more basic compared to the stomach, where the stomach is very acidic. Things like that contribute to what type of tissue is formed and what cells are located where in that environment. And then you have organs, conglomeration of the tissues, and then organ systems and organisms. Alright, moving right along. So I talked about what molecules are, cells, cell structure, definition of tissue, alkalosis, acidosis, end product of mitosis. I talked about energy production, and since I referred to where cells are located, I might as well talk about the positions of the body.